So first of all, good morning, good afternoon from Zagreb, from Croatia. It's very well into afternoon here. I was very honored to be invited to have this talk. Well, actually, I was hoping that Roxana will talk about marine megafauna and association of the diatoms with that, because it's a perfect intro of the lecture that I will be having. But since she's having the lecture after me, well, it will complement her lecture. That's the point, because she will talk about a lot more about animals who carry around different jungles on their back. Sea turtles are known to carry around the universe. We have the myths throughout years, and they all myths in the various cultures, various human cultures, are talking about turtles, not exactly sea turtles, but turtles who carry around our universe, the whole universe on their back. And well, the myth about the Discworld, the story about Discworld is from Terry Pratchett. I'm sure that most of you have heard about it, but he had the inspiration from old myths that depicted elephants as standing on the carapace of the sea turtle and carrying around this flat earth on their back. Well, it's not earth, it's the complete world. So we have this idea of having the whole world on the turtle's back. It's the turtles, the sea turtles that are inhabiting our world today. There were many of them that are extinct now. They've been around for millions and millions of years in our seas. And we have today the 7 extant species, which all more or less carry various loads of organisms on their skins and on their carapaces, and on their external surfaces, just, well, like all the other megafauna, because they're just like hard surfaces for the organisms to attach in these vast areas of the marine realm. So I'm not going to mention actually anything about other species of sea turtles, although there is plenty of things to say, but I'm going to focus, as I put in the introduction, on the loggerhead. Loggerhead sea turtle the name comes from its big head. Well, it's the head that you can see also here. The head is big and the jaws are very strong because it eats different types of organisms, various invertebrates, and it crushes the mollusks and different types of invertebrate, not just skulls, but exoskeletons and so on, and sponges. So it's an omnivore species, and it digs around the marine environment, neritic environment. It was described by Linné in his Systema, and along the description of Caretta, it was also the green turtle, this one. Where is it? Helonia midas. So they were described by Linné, but along this description, and well, not description, the taxonomy, the name, it was also described Helonibia testudinaria, one of the most studied epibiontic barnacles that is living on various types of marine megafauna, not just sea turtles. So Linné has recognized, Linné has recognized this as a separate species, and also different epibiotic species were studied first by Darwin. He was, I'm sure that you are well aware of, that he was very intrigued for years by taxonomy and physiology of barnacles. and studied in most details. And it was this fascinating way in morphology and physiology and the terms of reproduction that actually contributed a lot to his evolution theory. So the sea turtles carry around the whole complex set of organisms on their backs. You can see by naked eye loads of them, macroepibionts. You can see here, so this is a sea turtle from the Adriatic Sea, from the Mediterranean, that has these barnacles. There are several species of barnacles. Some of them are species specific to sea turtles, but not all of them. And they are mainly thought to be commensalic species. Several of species can harm sea turtles, so they here consider them as parasitic species. Several types of also barnacles, but other species. And on the sea turtle surface, we have plenty of something that we categorize as opportunistic taxa. The main thing, well, the main focus to study what is actually living on sea turtles is to differentiate between commensalic species and opportunistic taxa. And each of the group can tell us different stories about sea turtles, because they are like the complete world is on their back. So loggerhead sea turtles are living all around the world. You have plenty of them in the United States, and the part of the United States that is here, I mean, this belongs to the Northwest Atlantic Regional Management Unit. It's not common that Atlantic sea loggerheads get into the Mediterranean, but it happens sometimes. But most of the Mediterranean population is quite isolated from the rest of the world. And we in the Mediterranean don't have many of the species of the sea turtles. We have just 3. Leatherback is sometimes occasionally visiting, and we have nesting and reproduction. So reproduction and foraging for 2 species that actually Linné described. So the green turtle and the loggerhead sea turtle. The loggerhead sea turtle is estimated to have the largest load of epibionts, so number of taxa on its back and, well, on its carapace and on its skin. So as you can see, this is from the paper from Robinson and Faller, that most of them are belonging to this Malacostraca. So these are the barnacles. Where well, it's different, the load, of course, of the epibionts between different stages, life stages of sea turtles. The sea turtles, I'm sure that you all know that they are they're laying eggs, and after several 2 months of incubation, they hatch out, and then this happens on the beaches all around the world, and it needs to be the beaches needs to be sandy, because then the hatchlings migrate in the sandy beach. They migrate to the oceanic phase, where they remain between 4 and 19 years. It's the estimate, but it depends on the area and also on the population where the sea turtles are living. So subadults are mainly the organisms that are living in lagoons, estuaries. So these are younglings and, well, teenagers, I would say. And the adults that became sexually mature, they go again to the seasonal breeding, migrations, and so on. So the different stage of the life cycle also determines the epibionts that live on the turtles. I said that in the Mediterranean, it's quite a separate population, and the estimate that most of the nesting sites are actually of the loggerheads are situated in the Eastern Mediterranean. And the foraging area, as you can notice, so this is the area where we work and where the samples we have from the Adriatic Sea. In the Adriatic Sea, there are no I would say permanent nesting sites. There are occasional nesting sites in the western part recorded during last few years. And this is the place where the loggerheads come and forage, eat, and probably have fun, but not reproduce and not lay eggs. So during the last few years, so it will be, well, soon 10 years now, we sampled, well, yeah, the first samples we got in 2015, so it's like 10 years now. We scraped some sea turtles inspired by Roxana and other people discoveries of specific organisms, diatoms that live on sea turtles. So we've been scraping the epizoic biofilm samples. I said the Adriatic Sea, and you can see the locations where we took samples from. We focused mostly on the rehabilitation centers because it's the most convenient way how to get the turtle, the good sample from the turtle, because you have it in the, I would say, controlled environment, as you can see here on the table or in the rehabilitation tank. And also they're protected, and you need the cooperation with the people who have permits that who can actually touch and take samples from the animals. And we've been collaborating with various rehabilitation centers, and it includes also University of Bari, where they have the specialized sea turtle clinic, but also the Archelon Greece, which we didn't have the samples of from the rehabilitation centers, but they were generous enough to get us samples while they were tagging turtles. So these are samples that we have from one feeding ground. This is Ambrakikos Bay, which is the monitoring program for sea turtle is going on for now like 20 years, and they've been regularly taking measurements and sampling sea turtles. And also on Retimnos, which is our only nesting site of that we have samples from females that got out from the sea and laid their eggs, and while they were doing that, volunteers were sampling skin and carapace for us. So you can see also in the picture how the samples were taken. Most of them were taken by a very useful tool. It's a toothbrush that you scrape all kinds of biofilms from all other surfaces. And I won't present you all the results that we also have from the endozoic microbiota, because this is something that we were also interested very much about. What is the microbiome of the sea turtles? So the gut microbiome. But we scraped with a toothbrush, well, parts of the upper parts of the sea turtle, mostly focused on the places around their necks, but also flippers. And the carapace, it was after some time and after, some of the preliminary studies, we decided to scrape as much as we could. So we know today that there are loads of subhabitats on the turtle skin, on the turtle carapace. When I say subhabitats, that means that all of these large epizoic macroepibionts, barnacles, and also algae, marine algae, they all have their own microbiome and microbiota. So it's too difficult to separate what is actually on the sea turtle and what is on their macroepibiont. But we tried to take samples directly, scraping from the sea turtle carapace, the surface of the carapace. And when it was possible, these were the samples that I loved the most, because turtles are shedding their carapace, the scutes. Each of the sea turtles sheds their carapace scutes in a different way. Loggerhead does that in like complete peeling off the upper part of this carapace, the layer of the keratin, so you can get a very nice biofilm, which is actually the intact biofilm, how it was on the sea turtle, and this is like it's a keratin, so they do it normally. How often, I'm not exactly sure. They say they shed their carapace at least once per year, but yes, they're trying to get rid of everything that is growing on them, so it's not too much. One of the first efforts to study the microbial jungles was to get everything from these scrapings, and well, isolate the complete DNA and do the metabar coding via the 16S and 18S. So what prokaryotes are living in this biofilm, and also what eukaryotes that live in this biofilm. And the results were, well, a lot. And loads of taxa, and like I said, it was kind of a first thing that we wanted to do, is to try to separate what is actually the taxa that we can consider as commensal species, and which are the occasional hitchhikers, so this opportunistic species that come from the environment, but they would live anywhere else if something that they can attach to would pass by in this in the sea. And so in the prokaryotes, as the phototrophic biofilm was really interesting thing to us, we focused on the cyanobacteria and the diatoms, of course, as the main component of the photosynthetic microeukaryotic assemblage were our focus. So this was the overview of the surface microbiota, and as the cyanobacteria, the prokaryotic component, we wanted to see how many of them is actually in this biofilm of the sea turtles, and it it wasn't a lot in comparison to other prokaryotic genera, so we can't say it's like the biofilm is dominated. 16S component is dominated by cyanobacteria. The most of the percentage that it get, it's like 15%, but it's just, well, it's mostly in this Greek samples. We have some ideas why. And the samples that we had from Croatia had the most of like 10% of the cyanobacteria in the prokaryotic assemblage. On the right, you can see nicely how this biofilm looks like when you take a look on this scute that is shed from the loggerhead carapace. So the surface is not the filter, is the keratin from the actual keratin from the carapace, and this biofilm is formed by the interaction of the APS and the bacteria, and of course cyanobacteria that produce a lot of loads of the mucopolysaccharides and polysaccharides, and everything glues up and sticks together in this form. The core commensal cyanobacteric taxa, it was a hard thing to, well, to see. Well, we wanted to see if there are some taxa that will appear only on sea turtles, and these are the samples. P and R are samples from the pool, rehabilitation pool that the turtles were in, and R is the environmental sample. And well, it was most of the taxa, well, the genus that appeared on the sea turtles were also present in the environment. And that also includes the plastic in the turtle tank and also in the environment not connected with this sea turtles. But investigating of the cyanobacterial community, its diversity, when we compared different ages of the sea turtles, so we grouped sea turtles between juvenile, subadult, and adult. Juvenile sea turtles had the cyanobacterial diversity much less diverse. The community was much less diverse than the subadult and adult. And this is the measure how we measured the age of the sea turtles wasn't, of course, by a questionnaire. You cannot ask the sea turtle how old are you, but you need to measure its carapace, and there is a certain line of carapace length which says to you if it's which age group it belongs to. So the diversity is definitely significantly associated with this carapace length. And that's also connected indirectly with the age group that it belongs, that the turtle belongs to. So we just recently published this as an article, Growing Older, Growing More Diverse, as a title, because this is the main punchline. And we grew, tried to isolate and grew this cyanobacteria, of course, in the lab, and the most successful strains were, of course, the ones that belong probably opportunistic taxa that can grow anywhere, but some species of oscillatoriales, and this was identified similar to Blentotrix, so two of the strains. We have several of the floats of down these strains of cyanobacteria, and one group that we have several strains from, we can now hypothesize that it's not, as you can see, typically cyanobacterial greenish, blue-green, but it's going more into this pink direction, and it belongs to the Leptotoe, similar species, and our current working hypothesis that this is one of maybe commensalic sea turtle epibiotic cyanobacterial taxa, because this didn't grow from anywhere else that we had samples from, and it's also forming in the huge tree that you can see here. It's forming, these strains are forming quite a distinctive clade. So what's the relationship between epizoics and the host health? We know that from the macroepibionts, when the sea turtles look like this one, as you can see here, it just is totally ill. This one was really badly hurt with a bad head, but they can look even really, really much worse. It's something that is called debilitated syndrome, and it's usually connected with a heavy epibiont load. That means that the barnacles are completely covering the sea turtle, and definitely have the impact on their movements and so on. So this sea turtle that we had in our study, we sampled it 3 times and followed it how it recovered through 3 years being in the rehabilitation center. When it came, it had heavy epizoic load, but also under the microscope Bart and Van den Bever and the colleague from Van Tink, she was visiting Bart when I was there with the samples under the microscope, and we identified the Chrysophyceran stomatocyst, which is totally unusual. Well, our hypothesis is that this is something that got entangled in these epibionts, so the macroalgae that are growing, that were growing on this sea turtle, and accidentally ended up on this sea turtle. But that's also again proof of this microbial jungle. Really, if you take a look of the sample under the microscope, you can really find out loads of very interesting things. So finally, diatoms. Again, the question, which are the opportunistic taxa and which are the core microbiome, the commensal actually taxa, and throughout our studies, we called them putatively epizoic, so living only on animals. And they are, first of all, community. In our sampling effort was differed between the turtles, carapace of the turtles, diatom community, differed between sea turtles that just came in this rehabilitation center. So these are the turtles from Pula. And we also took samples from the environment, and Margot, she did the analysis, and these are communities completely different from the ones that we are finding on the sea turtle. But the carapace of the sea turtle is loaded with the diatom community that is very, very similar with the diatom community that we find on the biofilm that is growing on the sea turtle tank. This is a similar thing to the cyanobacteria. So these tanks are not cleaned daily, and the biofilm forms in the a few weeks, well, a week or two, or even a few days. So we find some of this species, and this is our idea that these are opportunistic taxa that can invade, well, not invade, but they populate the sea turtle carapace from the seawater in the rescue center, so their immediate environment, and the community is changed when the turtles are spending a long time in the turtle tank. We have more data, so these were like several samples of the sea turtles, and we connected the days in the rehab with the sea turtle, with the diatom community. Both the carapace and the skin are grouping together, connecting with the when the sea turtle is more days in rehab, its community is getting similar to the diatom community that we defined on these pools. This is one exception of loggerhead that stayed like 2 years in the rehabilitation center, but this is not the same tank she didn't stay, and we didn't sample it from the same tank that we did here. So it was, when I sampled it, it was moved to a different pool on the island where it was getting ready to be released back into the marine environment. So it stayed there for like 3 months. And this pool, this rehabilitation tank, is not enclosed. Well, it's connected directly with the seawater, with the sea, and it really mimics the natural environment of the sea turtle. So its community, after 3 months being in that natural environment, semi-natural environment, with the direct contact with the seawater, the diatom community apparently reverted to the more natural mode, I would say. So which are these opportunistic species and which are the core species? From Roxana next week, next time you will hear a lot about different animals that are basically hosting the same similar species like sea turtle do. And one of the star species is here, and you can see it in here in red. This is the most abundant species on the sea turtle carapace when they're not in the rehabilitation center but in the wild. And this is a species in species of Pollina, of the genus Pollina. And that was actually identified from different species of sea turtles by Roxana and Bart 10 years ago. And we did also in 2020, you can see it here. It's the weight, it's D and E are helonical, and this is the F is the Medlinella. So these are, and this is Proschkinia, species of diatoms in red. We consider today as commensalic species because we find them really connected with sea turtles. All other turtles, well, all other species, they can come from the environment, and it depends really where the turtle was spending its time recently, and when did you get to sample it. It really changes in probably seasonal. We don't have the seasonal following samples, but that's something definitely that would be worth looking into. As a first thing that we tried also to see the diatom community, the difference between diatom communities, it was the study with Roxana's samples from South Africa back then, and also from United States, from Tom and Frankovich and Matt Ashworth, samples from Florida. And these showed the first differentiation between the diatom community, but it's not the answer if this epizoic, true commensalic epizoic taxa are differs between these populations of sea turtles that inhabit different seas. What we also know today that they are the community of the epibionts. It's not just for the microepibionts, it's also from the macroepibionts, for the barnacles. Different species you can find on the skin and on the carapace. It has many plausible explanations and hypotheses, but for instance, let this under F is Medlinella, the species that Tom Frankovich had described from the neck of the loggerhead sea turtle back in Florida, and it was one of the most, I would say, often encountered species in the skin samples that I also had from the Adriatic Sea. So, of course, I said there are differences between carapace versus skin, and the carapace community, like all other epibiontic community, is much more diverse, and the skin habitat, not just for the diatoms, but especially for all the epibionts, is much more selective. So they're not, the diversity is not high, but it's more populated by more commensalic species, I would say. And we tried to see if there are some differences between our communities of sea turtles between in the Adriatic Sea, if we can see some geographical location separations, but it's definitely something that was not clear. And unlike the cyanobacteria, well, also age group, it didn't show any particular grouping of the community, except in the Archelon samples that we had from Greece, where, well, this adult, as you can see in pink, and the subadult animals in blue, but they are taken from a different life stage of the sea turtle. And as you may remember, I told you that we had those adult samples from nesting females, and they were sampled while they were out and on the sand, and probably didn't eat in the mud like these sub-adult turtles in Ambrakikos Bay do. So their community is something that got in our significant difference between age groups. But otherwise, the other samples didn't, the diatom community didn't have many differences between the stages of sea turtle. So I said Pulina, Medlinella also mentioned, and this is the and we also have some Proschkinia, and Pulina, you can see it's depicted here by Clara in green, and it's really, in when you take a look at this skewed in the scanning electron microscope, it's olive, everything is connected. So putatively epizoic taxa, this commensal taxa and opportunistic taxa, they all grow happily in this biofilm matrix connecting with their slime that they are producing, various stalks and so on and so on. As I can also, there are Nichia inconspicua here also in the picture, which is really a name for a large, I would say, group of organisms. I didn't get into it's a complex thing. So maybe some of these taxa are actually in that we call Nichia inconspicua, because we found them also in the pool sample. It's not really straightforward if it's commensal or if it's opportunistic taxa. But I said the skin is really, really interesting. And yes, this is one of the small projects that we had during this last years. In 2020, well, we took a look at several samples. It was just, I remember 8 samples, something like that. And it was the heads that we got from the turtle biologist that was giving us diatom people to scrape. And this was really the most convenient thing, because trust me, for the live, when the sea turtle is alive and kicking, you don't want to go near to this beak and sample with a toothbrush. Well, we did, because we took oral samples from the swabs, and it was really an adventure to sample this. But in this study, as these turtles were really dead, so for a long time, we also, and frozen for some time, we could take their skin not from nostrils and from the beak and everything. And when we take a look under the microscope, it was these are commensal taxa that are common for the Adriatic Sea. I have to emphasize that there are plenty of other marine animal epizoic taxa, Tursiocola, for instance, but it's not so common in the Adriatic samples. I just had few sightings, but it's not something that is dominating the diatom community like this does. So for instance, you can see here, this is a picture from Pulina, which is like full of one single species, the town that it forms, strictly monospecies. And in this Pulinas and Helonicolas, so it's a genus. You have several species, especially Helonicola, but it has this really, really variable morphological variability. And Pulina can be large. Okay, diatoms are definitely differ by size because of the life stage. We all know that, but it can really have different morphology. But it's so abundant on the normal type of turtle sample that when you start to work with these samples, you just recognize it immediately. And Helonicolas also are very, very abundant. They have a few more pores in the areoles here, and that's the difference between Pulina. So also in the last years, we have been describing new taxa from the turtle samples, but I have to say now that most of them, well, that all of them described from the Adriatic, it would probably be categorized as opportunistic diatom taxa. For instance, Maevskaya istriaca, we know now that it's really common in the aquarium samples, so in the biofilm of that grows on the aquarium tanks and so on. Pushkinia sulcata is still a little bit, well, fishy. From the United States, from Florida samples, Matt and Tom had described Vergo striata, which is more epizoic also in the Adriatic samples, but sulcata can also be found in the sea, around the sea turtle environment, so in the different types of substrata, including plastic and so on. Catenula is also one of the species that we have described, but it's also something that can be found in the biofilm around the sea turtle. And species of Planotidium. We also described one of the species of Helocera here, and it's not that it's opportunistic of the sea turtle, but it's probably probably epiphytic taxa that is living on the algae that are growing on the sea turtle back. So lots of new species, but still after this time and all these investigations, I'm really convinced that these are the opportunistic ones and are not the really core diatoms that are connected only with sea turtles. The ones that are, so Pulina, that you already saw, we of course isolated strains from the Adriatic. We had just several strains, but Matt and Roxana and Thomas and all the other people who contributed, there are plenty. You see here Terciocola. Probably Roxana will mention more about this genus. Like I said, I have like 2 sightings of this in my samples, and Medlinellus, and different hosts are having different epibiotic diatoms. Some of them, like in other hosts, are shared between different turtle species, but some of them are more specific to the sea turtle species, to each sea turtle species. So Pulina is, this one is really abundant in the Adriatic loggerhead sea turtles. And again, as I say, if I take a look at the samples that is full of Pulina, I would say, yes, that's come probably from the Adriatic. Well, it doesn't have to be, but this is a healthy sea turtle that was roaming around in the sea until it was sampled. So for you, the resource that we provided from the project and from all these epibionts, it wasn't published as a paper, but it's free for you to use. On the ResearchGate is the poster that was presented in the last European Diatom Symposium in Ohrid, Macedonia, and they are all strains are deposited in gene banks, so that in the gene bank and available, that are so the opportunistic taxa and that we isolated, and also the epizoic taxa that we isolated. We tried to have for all of them 3 genes. And interesting here is Achnante's story that is developed currently by Matt, and we have plenty of strains from the Adriatic, and it's kind of in the 3 genes phylogeny grouping together with all other strains from the rest of the sea turtle species, but also from the different environments that it was isolated from. So I mean different populations of sea turtles, of the loggerhead sea turtles. And this is the story that we are working. So if you need to use and access, the whole information table is available on the web page of the project. I hope it will go on for some more years. If my university doesn't cut it in next few years, I will try to keep down. Okay, it doesn't exist. so it doesn't bother anyone. So it will be accessible here, and also the full phylogenetic tree. And if you have any questions, the strains are not alive in, I think we have some of them cryopreserved in Belgium, in Ghent, and some of them are maybe in our lab, but I'm not sure in which state they are. But all the information that you need, you can just email me and ask me if you need anything more about, and also the all the pictures that we took from all the strains. So LM scanning, live, whatever do we have. So this is the resource. I said to you that the Paulina is really beautiful, and it was our interest to see also the bacteria that were living and isolated together with the diatoms in this culture, in the cultures. And this was one of my PhD student work. Clara, she also put a really special attention to the bacteria that were isolated with these strains of Achnanthes, of Paulina, but also the Entomones, Phalatia, Niches, or something that we consider as opportunistic, and to see if there is maybe a difference between the bacterial community that is growing with the epizoics, so with the commensal and with these hitchhikers, well, the opportunistic species. And well, it was quite also an experience in this microbial investigation of microbial jungle, because apparently that cultures enrich the taxa that are scarce in the original habitat. They just come in the better adapted environment in the culture. So enrichment culture technique, it really gets us some of the taxa, for instance, Alcanivorex, or Flavobacteriaceae in the bacteria. It's not strictly connection with the diatoms. They're just living probably in their phycosphere, but they are enriched in the culture while it's while the selection process is going on. And also, but there is group of bacteria that is connected with the epizoics that are in cultures, and these are Phylum Plankton Mycetota, and it's not, you can see here the Achnanthes, and growing these stalks, loads of APS, and these are bacteria that are probably associated with the large amounts of APS in the culture. So it's their source of nutrition, food, and whatever. So it's their enrichment that gets them their population going, and we found them in Paulinas and Achnanthes species particularly abundant. And in the end, I would like to say, of course, thank you for listening. And yes, I'm contributing all the people who contributed to this work, and I'm open for questions.
Well, thank you, Sunc, for that interesting presentation. If folks would like to ask questions, we've got a small enough group right now that if you feel like just raising your hand, we can call on you like that. Or if you want to, you can also type a question in the chat. As a starting point, one of the things you brought up right at the beginning was that among the turtles,
Can I stop share or what?
Oh,
right. Tell me.
The greatest diversity of epibionts on turtles was on these loggerheads, and I'm just curious, yes, why they win. There it is.
Well, yeah, there it is. Well, probably because of their habits. The green sea turtles are herbivores, and they're like cows. They eat mainly on algae, and this is not the way how they are getting their food. Loggerheads with their big heads, they dig in the mud. So if you can see maybe here this is a sea turtle that was brought up from Ambrakeikos Bay. This is a typical teenager eating everything that it can, get its beak on, and getting into the mud. So the samples that I got from Ambrakeikos Bay, but not just that, in also these neritic areas, they go deeply into the substratum and they just get everything on them very easily because they are
Okay.
in direct contact. So other species, some of them are eating just sponges on coral reefs, you know, just they're not in the mud, in the substrate. Loggerheads have, all of them have these habits, but so the juveniles probably have less load. Not okay, they're juvenile, but also they are eating in the ocean and in the pelagic environment. So not digging in the mud, they're eating, you know, the zooplankton and some of things that they have. So this is one of the explanations that is maybe probable, but yes, it was recorded for the loggerheads, not just the Mediterranean, but like the species is full of epibionts.
That's really neat. Anyone have any questions they'd like to ask? Oh, Marina, I saw you raise your hand. Go ahead.
Yeah, can you hear me?
Yep.
Yes.
Yeah. Thank you very much, Sunc. Very, very interesting talk. I was wondering, so you mentioned that cyanobacteria are more diverse on older turtles, right?
Yeah.
Is that true also for other groups, taxonomic groups, or it's just cyanobacteria?
Well, we study cyanobacteria, but that's also something that is our hypothesis. It's connected with the age of the biofilm that is formed. So when you have a juvenile, it's you know, the biofilm just starts to grow, and it's not sub niches for all these epibionts. It's not so diverse. It's pretty much the juveniles really have this smooth carapace, and then you have just few bacteria, and then this the epibionts. Yes, so I have found on sea turtles that were like really 20 centimeters, like it's really small. I have found Paulina, so but not cyanobacteria, because they probably come into this biofilm, and all the macroalgae also come, and well, it's this development of the biofilm that is.
So so you mentioned that they actually shed this upper layer, right?
Yeah.
So how?
But it's not a complete. It's not like crustaceans or something. It's not a complete shedding. They tried to get rid of the epibionts. They are scraping their backs. They are shedding this, but they can, you know, they shed 1 or 2 scutes. The rest is there, but then you have the growth of the biofilm. It's very fast.
I see. Okay. So these barnacles, they're like sitting there forever. It's not just like they grow that fast, right?
No, they sit there for some time, and they can move. So you have beautiful it's videos. So here, this barnacle, they had made the video how the barnacle actually travels around the turtle's head. Yes, these are animals. It's different. But also the diatoms. When Paulina is very active when it comes, we saw it in culture, when you isolated it, it's not, you know, a stalk immediately. No. It's very active, trying to go around and find a place where, and then it forms stalk. Achnanthes also. So they're trying to find their place on this world.
It's pretty much like Dolphinimus do, you know, the same. So they crawl and then they start. Okay, great. Thank you so much.
Oh. Let's wrap up with a question from Matt here. Go ahead, Matt.
Hi, Matt.
Uh-oh, do we lose Matt? There he's
no,
coming.
No, still here. Sorry,
Matt.
Sorry. Good to see you again, Sunc.
Yeah.
I know we're up against noon here, so I will try to keep it short. But one thing I did want to ask you about is particularly looking at those bacterial assemblages from the cultures of the putative epizoics. Did you compare those to the natural assemblages you found on carapace versus skin?
Yeah, carapace versus skin. I'm not sure. We compared, so the assemblages the bacteria that were found in the isolated. Wait, I didn't put this slide here. And in the primary isolated, the samples that we isolated from, but I'm not sure. So these are down there, TB, whatever. Let me see. No, none of them. They were all carapace samples.
They're all carapace samples.
Yes, because
Okay.
the skin samples are pair numbers. I know that.
Ah,
Sorry. Yes. So
All right.
these were all the carapace samples because they were kind of, well, it wasn't intentional. It was just that these strains that Clara had grew from carapace samples better.
Okay. Yeah, because I mean, obviously, one of the big questions here is, you know, what makes them commensal or endemic or putatively obligately epizoic when we can pluck them off the turtle and grow them just fine in tubes and petri dishes, right?
Yeah, but that's the
Um,
point, and I think it's the most well, I've been studying this epibiont literature. It's just like all the other epibionts do that. You have some
Yeah.
don't have acid, this special stuff. You know, they can survive in this environment, but they kind of flourish on the turtles. It's not just they would die because of it. Maybe in the evolution. I mean, that's why there are in all the species of the sea turtle present. But yes, bacteria, I think they had, it needs a special attention. It's much more difficult to draw some conclusions than the diatom communities and even for the cyanobacteria, because bacteria are connected with all of these micro-eukaryotic species have their own microbiome. So like we are studying the macroepibionts, like I said in the beginning, you know, that Roxana had found the specific species of diatoms connected with sea barnacles, with the barnacles. So it's this hierarchy of habitats, and this is the same thing with for the bacteria. I think it's to try to find, it would be interesting also to see the skin bacteria especially, but I don't know, we didn't use the skin samples from our for the 16S studies. I discarded it because I went to the phototrophic biofilm, to tell you the truth. So it was like, yes, it's developing on the carapace better to have more sea turtles than to have the sea turtles, or the same sea turtles, the skin and the carapace.
There you go. Well, that sounds like you got your next proposal ready to go there.
Matt has enough things.
All right.
Well, so do you, I guess. But
yeah.
Well, we've reached the top of the hour, folks.
Yeah.
Thanks again for joining us for another Diatom Web Academy. I want to especially thank Sunc for offering up such interesting data and interesting observations on sea turtles. Please, everyone, join us again next week. Be 1 week away only on I think it's Tuesday, April 1st.
Yeah.
And Roxana is going to be speaking on epibionts on many other of marine megafauna. And I think we're going to see a lot of connections to the sea turtles, but I bet we see a lot more stuff too. So anyway, I appreciate everyone joining us today. We'll have this recorded, and probably within about a week, we'll have it up on our YouTube channel. Feel free to also find all the other Diatom Web Academies on the YouTube channel. If you can't find a link to that, go to diatoms.org to the Diatom Web Academy link we have there, and you can find links to all of those things. So thanks again, Sunc, and good to see everyone today.